Welcome to Hope One, ladies and gentlemen. We are live this morning, 11:30 a.m. Eastern Time, 8:30 on the West Coast, and we have a program today that you're going to enjoy, and we're going to have time for questions at the end because we're talking about viruses and intelligent design. And there are no two better people on the planet to talk about this than my friend Dr. Stephen Meyer and my new friend Dr. Scott Minnick. Both are experts in this area. You know, Stephen Meyer has been on the podcast and has written several books. Of course, he's written Signature in the Cell and Darwin's Doubt. He has a new book coming out next year called The Return of the God Hypothesis. Dr. Scott Minnick is a professor of microbiology. He teaches at the University of Idaho, and he's coming to us as well today. He's also affiliated with Discovery Institute. And both gentlemen are coming from the West Coast. Let me bring them on right now. Here is Dr. Steve Meyer and Dr. Scott Minnick. Gentlemen, good morning.
Good morning, Frank.
Thanks so much for coming on to talk about this issue, because people are wondering what a virus is, and they're wondering how this comports with the Christian worldview. But before we get into viruses, Steve, I think we really need to start with intelligent design. What is it?
Well, intelligent design is the idea that there are certain features of the universe and of life that are best explained by reference to designing intelligence or a mind, rather than an undirected, unguided material process. And the kinds of features that we look at in formulating the theory of intelligent design, or the evidence that we're talking about, are things like the digital code stored in the DNA molecule, the irreducibly complex circuits and machines that we find, little nano-machines that we find in living cells, but also in the area of physics, what's called the fine-tuning of the laws and constants of physics, and the initial conditions or the setup of the universe from the very beginning, all bear evidence of a designing intelligence. The physicists talk about fine-tuning. Intelligent design would infer from the fine-tuning a fine-tuner. In biology, we see evidence of a digital code, one that, in fact, Bill Gates has said is very much like a software program from which we infer a master programmer for life. So that's roughly a quick sketch of the idea that there are things in nature that bear witness to the reality of an intelligent agent behind nature as intelligent cause.
Scott, Steve's work has done a lot of work, quite obviously, in the fact that there's this genetic code and there's a source of information in living things. You've done a lot of work in this idea of irreducible complexity with your colleague Michael Behe, also the Discovery Institute. Can you tell us a little bit about what irreducible complexity is?
Sure. I think it's really, I think it's the bread and butter of molecular genetics in terms of dissecting the nanomachines that Steve referred to and figuring out how they work. I mean, we don't have the blueprints. So essentially what you do, say if you're looking at the bacterial flagellum, is working with E. coli or Salmonella, or in my situation, Yersinia, you have to make mutants. You isolate mutants of bacteria that can no longer swim, or they can no longer chemotax. They can't sense their environment. You map where these mutations are. You determine how many genes are involved. And so all of the components for the flagellum have been identified by this loss of function. If you knock out, the clutch, if you knock out the U-joint, if you knock out the propeller, if you knock out a stator, these all have effects. And so by this reverse engineering, we have figured out that there are about 30 key components for this very elegant rotary engine that are all necessary. And so when you look at the constraints on these nanomachines, how these parts interact, then from a Darwinian standpoint, you have to say, well, how did these come about in a stepwise manner that's required by mutation and selection? And it becomes very difficult because we don't have the antecedents for the individual steps of this process. We know that if you knock out one component, then the whole system has no function And if it has no function, you have nothing to select. So that's the basic, I think, underpinning of irreducible complexity.
Now, Steve, how have the Darwinists responded to irreducible complexity? Have they given any robust answer that can be demonstrated to be true?
Well, they've posited the existence of a precursor system called the type 3 secretory system. And it's a little syringe-like device that in an ordinary flagellar motor it sits on the inside of it, and then the other parts are built around it. But in some bacterial systems, you have the type 3 secretory system existing on its own without the rest of the parts. And so the thesis is that that type 3 secretory system, the little syringe or needle, was the precursor. And it's got, Scott could tell you exactly, but I think it's 10 to 12 proteins in most systems. But you got to get to a 30 protein system in the flagellar motor. So if you take what Scott just said and you think about the 29 part, the 28 part, the 27, 26, 25, 24, 23 part version of that flagellar system, none of those alleged or hypothesized intermediates form any function whatsoever. And so there's a huge difficulty in getting from a type 3 secretory system, even if it is the precursor, to the fully formed flagellar motor, because those intermediate stages, again, have no function. And yet Darwinian evolution works as functional advantages are preserved and then passed on to the next generation. But if there's no function, there's nothing to preserve, nothing to pass on, and those intermediate stages would not be viable. So there's still a huge gap. But beyond that, we've learned something really important from what are called mutation density studies and some other lines of evidence, and namely that little syringe-like device seems to be a devolutionary breakdown product of the complete flagellar motor. In other words, the flagellar system as a whole, when it's found in isolation, is the genes that make up the parts for that system are older, not younger, than the genes that we find that make the type 3 secretory system when it's found in isolation. So either the type 3 is an independent system, or it's something that has devolved from the more complex aboriginal system. And,
Scott is. Oh, sorry. Go ahead. Complete the thought.
Yeah. So in any case, it's not the precursor, and therefore this hypothesized or proposed scenario in which the flagellar motor built up gradually from this simpler system is not credible in terms of molecular genetics, or theoretically credible when you think about what would be needed to get from that system to the whole flagellar motor.
Scott, is the type 3 system itself irreducibly complex?
It is. Again, it was defined by mutations and identifying the parts that lost the ability to, virulence factors or anti-host factors. And I think, again, coming back what Steve said, we find flagellar systems across all the phyla of bacteria. Type 3 secretory systems are in a very narrow range of, the gamma proteobacteria. And it's really interesting that, Mike Conkle at Washington State University works on a pathogen that we call Campylobacter jejuni. And, from the way it caused disease, that it had a type 3 secretory system, but the genomics said no, all it has is a flagellum. But it uses the flagellum for both motility and, if it encounters the mammalian host, to use the flagellum to inject these anti-host factors into our cells and cause disease. So there you have the precursor for the type 3. The flagellum's doing both functions. And you can see where perhaps through degradative processes that the secretory mechanism of the flagellum could be usurped for other than causing motility in a cell.
Steve, how many, maybe a hard question to answer, but when we look at the bacterial flagellum, that's just one example of an irreducibly complex system. How many systems are inside a living thing that could be considered irreducibly complex? I mean, just because you might come up with a possible way of explaining it naturally, one irreducibly complex system doesn't mean you've explained all of the irreducibly complex systems. So how many different systems in a living thing would be irreducibly complex, or could be, or appear to be?
Well, first I'd stipulate that no gradual Darwinian step-by-step processes have sufficed to explain any irreducibly complex system.
Mm-hmm.
So I wouldn't concede even the premise of the question.
Mm-hmm.
The best such proposal that has been made has been this type 3 secretory system as the precursor. And I've only touched on a couple of the problems with that proposal. But it turns out, I mean, this is one of the big discoveries of modern biochemistry, molecular biology, molecular genetics, is that at the foundation of life, you have many multiples of systems, various types of nanomachines, circuitry, information processing systems. I've worked principally on the question of the origin of the genetic information that's necessary to build the proteins and the protein machines that keep cells alive. And therefore, that's a question that strikes right to the heart of the question of the origin of the first life. How do you get the information that's necessary to build even a simple one-celled organism? That has turned out to be an insoluble problem in origin of life research. Even leading people in chemical evolutionary theory themselves acknowledge that we really are nowhere on explaining that. And yet we know from experience that whenever we find information, especially when we find it in a digital form, as we find it at the foundation of life in the DNA molecule and RNA molecules, whenever we find information and we trace it back to its source, whether we're talking about a hieroglyphic inscription or a paragraph in a book or information embedded in a radio signal, we always find that information arose from a mind, not an undirected process. So that's sort of the foundational argument for intelligent design. But then that information makes these irreducibly complex systems. The information encodes for their construction through very complex kind of assembly routines that themselves have a kind of irreducible complexity to them. But anyway, back to your original question, there are many of these irreducibly complex molecular machines. We have several animated on my website. The ATP synthase, the machine that makes the ATP molecules that store the energy that all cells need to power metabolic processes and reactions. There are robotic walking motor proteins, kinesin, and they walk like on two legs on these long tracks that are essentially functioning like railroad tracks inside the cell for all the automated kind of factory functions that it performs. And the kinesin tows these vesicles along the tracks, and the vesicles contain materials that are needed at other places within the cell. There are turbines. There are in the process of DNA replication, there are little machines that look like sliding clamps. The biology at the molecular level is full of these sophisticated nanomachines that are clearly accomplishing even mechanical processes that are discernible to a mechanical engineer. The flagellar motor is fascinating because it has, as Scott mentioned, a rotor, a stator, U-joints, bushings, a drive shaft, a clutch, on a miniaturized scale. Some of the systems that Scott works on can change direction on a quarter of a turn and rotate at 100,000 RPM. They've been said to be the most efficient machines ever discovered, yet they're in the cell membrane of a one-celled bacterial organism. So I like to say they're high-tech and low-life. So this has been a huge revolution in molecular genetics, in biochemistry, molecular biology, the discovery of this nanomachinery at the foundation of life. And it complements the discovery of the code, because just like if your viewers are thinking about an analogy to computers, if you want to give your computer a new function, you've got to give it new code. And the same thing turns out to be true in life. If you want to build a molecular machine, if you want to build a circuit, and there's all kinds of circuits that control animal development. If you want to build an information processing system, you've got to have code. And so that you want to build the first life, you've got to have code, just like in our computer world. So that's the ultimate question. Where does that information come from? But we also see these striking indicators of design and engineering in what the information builds, namely these nanomachines and circuitry.
I think that's what some people really don't get, Scott, is that in order for there to be natural selection, there's got to be something to select, correct? I mean, where does
Correct.
how does natural selection work? What happens?
Well, I mean, that's the question. How, is it a positive process that you can start with something simple and make it more complex? I think the opposite is probably true. When we look at organisms, and there are many examples now that were at one time modal. They have the genetic information to build a flagellum, but they've acquired a mutation. And what you find over time is the system is even further degraded. So it's a disadvantage for some organisms, particularly pathogens, to make flagella in a host, because we're looking for some of these proteins. And so it would be an advantage to shut the system down by mutation. But then when you see what happens over time, is the other genes downstream also become sinks for, insertion elements or other mutations. So if you don't use it, it degrades over time. And that is, antithetical to the process that you need for some antecedent product to be held in place so that you can add to it sequentially by mutation and selection. It just doesn't seem to work.
Yeah,
We can't have
we
it
should
both ways.
stipulate, Frank, that ID proponents all accept that mutation and selection are is a real process, a combination process, and both things are at work, natural selection and mutation. But what we see is that either they preserve very modest changes that slightly optimize an existing fundamental unit of life called a protein fold, or they degrade. We do not have evidence of mutation and selection accomplishing fundamental innovation, building new protein folds. So you can slightly optimize ones that exist already, and you can degrade genes and the proteins that are coded for them. So it's not a process that you can invoke to account for the large stepwise or abrupt increases in genetic information that we see arising in the history of life. And that becomes a big problem when you look at events like the Cambrian explosion, about which I've written, or the mammalian radiation, or any of the numerous abrupt appearances of new biological form, or what's biologists sometimes called morphological innovation. In 2016, a number of us, or 18, was it 16? Yeah, sorry, a number of us attended a conference in London held by the Royal Society, which was called by a group of evolutionary biologists who were very dissatisfied with the standard neo-Darwinian story about the history of life. And they're looking for new mechanisms of evolutionary innovation, because they recognize that neo-Darwinism and its mutation selection mechanism, as one scientist put it, lacks a theory of the generative. It doesn't explain major innovation. It explains, in fact, we heard over and over again biologists saying that mutation and selection can explain the survival, but not the arrival of the fittest. Explains how things are slightly modified to continue to survive if you can presuppose the form and structure they already have, but the mutation selection mechanism doesn't explain how fundamental innovation arrived in the first place.
I think it's important to point out to our audience now that when Drs. Meyer and Minnick are arguing that there is some sort of intelligence out there, this is not a God of the gaps argument. They don't just lack a natural explanation for the appearance of information or the appearance of these irreducibly complex systems. The appearance of information in irreducibly complex systems are positive evidence for an intelligence. They don't just lack a natural explanation. Of course they do, because there is no natural explanation, but there's also positive evidence for an intelligent being when you see genetic code or when you see an irreducibly complex system. Steve, do you want to go further with that?
Well, yeah, let me unpack that a little bit more, because we've been arguing that mutation selection doesn't explain irreducibly complex molecular machines, that random mutations don't explain how you get code. Random mutations, after all, degrade information. They don't generate. If you just think of that quickly, if you have a section of functioning software that performs a program for your laptop, and then you allow a series of mutations to the zeros and ones in that code. Ask yourself, are you gonna get a new functioning program or operating system, or are you going to degrade the one that you have already? And it's obvious when I ask this rhetorically, programmers get it. So we are lacking naturalistic accounts of the origin of information. They're not plausible explanations for that. But we also know of a cause which is sufficient to produce systems that have the feature of irreducible complexity. And we also know of a cause which is sufficient to produce digital code. And in both cases, we see that cause at work in our own world of high technology, namely intelligent agents are able to produce code. They're able to produce systems that are functionally integrated in order to perform some overall purpose. And so what we're seeing in these molecular machines is a systemic feature that we know arises from one and only one type of cause, and that's from intelligent engineering. Similarly, when we see the digital code at the foundation of life and DNA, we're seeing a type of artifact or type of evidence that we know from other experience arises from intelligent agents. It takes a programmer to make a program, in brief. So we're basing our inference to intelligent design on our established knowledge, not ignorance or gaps in our knowledge, but our established knowledge of cause and effect processes. That we know that intelligent agents can produce code and can produce functionally integrated, irreducibly complex systems. So when we see them at the foundation of life, it's a reasonable inference. In fact, an inference to the best explanation that a mind plays a similar role in the origin of these systems.
I want to ask Dr. Meyer and Dr. Minnick some questions about viruses in particular, since we're dealing with the coronavirus. And then we're going to take your questions. So if you viewers want to ask a question of Dr. Minnick or Dr. Meyer, type the word question in capital letters on the YouTube or Facebook stream. And in 10 to 15 or so minutes, we'll start to get to those questions. Just type them out clearly. Use complete sentences, succinct English, and we'll select your question if we hopefully can get to it. But let me ask Dr. Minnick, Scott, let's talk about viruses for a second. We're talking about in the context of the coronavirus. What is a virus? And then I'll ask Steve a question about, are they designed? So first of all, what is a virus?
Okay, virus is the ultimate parasite. It's these are right on the edge of the living and inorganic world. They're essentially what we say, bad news in a protein coat. It's a short genome that codes for its component parts. They don't divide like cell systems do. This little piece of information is injected into a cell. It replicates and then it assembles and produces progeny virus from that. So it's essentially a stripped down code. They're completely upon their host cell to use all of the machinery, the molecular machinery, the ribosomes, the zip code transferring system to get these proteins to the right place in the cell for assembly. And that's a virus. They can be RNA viruses like the coronavirus, DNA viruses, single-stranded, double-stranded RNA or DNA. So they're just short genomes to make more virus.
And are any of them beneficial to living things?
That's a question, and I wonder if our view isn't skewed just from necessity. In fact, when I go back to bacteriology, I'm more of a bacteriologist. The early history of bacteriology was studying disease because these are what affected us. It wasn't until, a decade or so that people recognized that bacteria are everywhere and they provide vital functions, in terms of the ecology of our earth and cycling of nitrogen and carbon and sulfur, and that the bacterial pathogens are such a small segment of the vast number of microorganisms that are playing a very crucial and important part in our ecosystems. Viruses, this is a newer field. They weren't discovered until the teens, the 1918, and we couldn't really work with them that well other than viruses until the cell culture was developed in the 1950s to produce polio vaccines. So I don't know. I mean, that's an open question.
Okay.
Do they serve an important part? We know in bacterial systems, they can be taxis of genetic information. So you can have a bacterium that acquires a mutation, and the virus can pick up, part of the DNA of the wild type bacterium's genome and transfer it and repair that lesion that's occurred, whether it's a point mutation or a deletion. So they can ferry genetic information between organisms, and that could be a beneficial role.
Steve, I'm sorry, Scott, go ahead, finish.
No, I'm fine.
Okay.
Hand it off.
Steve, are these viruses, are they designed, and if so, how do we detect that they are?
Well, I'm gonna decline to answer that for a minute and put your question in a broader context.
Okay.
Because Scott's been right in the middle of this as a expert in what's called virulence.
Uh-huh.
And I want to talk first about, let's just frame the question around natural evil, okay? Because this is what bothers people. If we got all this wonderful evidence of design in life, then what about the stuff that's not so nice? Okay.
Right.
What about things that look designed but also are harmful to humans? And the the biggest class of such virulent organisms are bacteria. And we've had, for example, the plague, Yersinia pestis, the system that Scott works on, killed hundreds of millions of people in the Middle Ages, and we've had outbreaks of it throughout human history. So what's going on with this phenomenon of virulent bacteria? So if everything was designed, why are there things that are bad design? And that's probably the most difficult question for proponents of intelligent design, but it's especially difficult if you hold to just sort of a secular ID perspective. There's a designer out there, we don't know who it is. We see evidence of design in nature, but what do we make of this stuff that looks badly designed, or looks maybe even good design, but it looks like it's very harmful? Both Scott and I are not just ID proponents, we're also biblical Christians. And so we have explanatory resources that maybe a generic ID hypothesis would not be able to access. Because if you look at Scripture, you see you would expect to see two things in nature. You would expect to see evidence of aboriginal design, good design at the foundation of life and in the universe, but you would also expect to see evidence of decay. In the book of Romans, it tells us that from the things that are made, the unseen qualities of the Creator are clearly manifest, Romans 1:20. But then when you get to Romans 8, it says that the creation itself is groaning for its redemption, and it actually says that the creation is in bondage to decay. And so if you accept the ID hypothesis, but within the framework of a sort of Judeo-Christian worldview where you're taking the scriptural ideas about the fall, something having gone wrong that has even affected nature, then you expect to see not one thing, not just good design, but evidence of decay of that design as well. And this is where conversations with Scott, for me, have been so illuminating, because it turns out that in his field of bacteriology, and that virulence, the acquisition of sort of a toxic function of bacteria vis-a-vis human beings, is invariably the result of mutation. It's the result of either the transfer of genetic information out of its original context into another organism where it's expressed out of context, processes that are things like lateral gene transfer and so forth, or else it's the result of the loss of genetic information. And fascinating, I heard Scott give this talk when we were lecturing in Brazil several years ago on the natural history of the plague. And the plague evolved from a very harmless bacterium that for which we have a natural immune response. That flagellar motor we were talking about, our immune system is all over that. It's got lots of bacteria. We have lots of bacteria in our gut that we need, and we have an immune response to the presence of the flagellar motor in those in those bacteria. But when you through a series of mutations, that harmless aboriginal bacterial strain, and we can actually track the mutations. Scott should tell more about this, but you can track them through human history. as you find these bugs in the pulp of teeth and things like that. And by the time you get to the Middle Ages, there have been 4 identifiable mutations that have degraded that original system, and that flagellar motor is now, the whole system is gone. All that's left is this secretion device that's at the core of the motor that's around which it's seated, and now it's secreting nasty stuff for which we had no immune response, and you get a nasty bug. Well, what is that? That's decay. That's decay of the aboriginal design, and it's exactly what you would expect on a biblical theology of nature, that you would expect to see evidence of the original design, but then evidence of decay. And then the irony is that the process that the neo-Darwinians or the evolutionary theorists, the process to which they attribute creativity, namely mutation, which is the engine of creativity and innovation in the mutation selection process, or at least allegedly so, the process to which they attribute creativity is actually the process that produces decay, and then what we call natural evil. So we can clearly see that in the case of bacteria, the nasty things it does to humans was not part of the original design, which is very consonant with a Judeo-Christian view of the natural world, and a Judeo-Christian understanding of how ID functions, or where it functions. It functions at the beginning, but then there's decay subsequent to that.
So Scott, we always hear evolutionists claiming that evolution creates, and evolution is evidence for, or natural selection, mutations are evidence for this creative process. Steve appears to be saying it's exactly the opposite, that it's a degrading process. Is that what you're seeing as well?
Well, I think it's through decay and selection. So maybe it's more of a preservative than a generative system. I mean, you can look at antibiotic resistance, right? In bacteria, you have a mutation that allows the organism to live in the presence of antibiotics that it didn't have before. But when you dissect what's actually going on, you've degraded part of the information processing. So a pretty significant fitness cost, but it preserves the organism, all right? So there's mutation, selection, but you haven't built anything new. You've actually wrecked part of the translation system or the DNA replication system that renders the organism immune to the antibiotic. Remove that selective pressure, and you often lose those resistant organisms pretty quickly because they can't compete out like the environment. I think in terms of what Steve was saying too, that's a surprise when the problem of natural evil and disease goes Darwin. I mean, Basic Gray. He says, I can't believe in a benevolent God that would create these parasitic wasps that would lay their eggs in a caterpillar, and their larvae would eat the caterpillar from the inside out. So you have a stinging wasp that deposits as host, and it looks horrible. But was that part of the original design? And we're seeing that it wasn't. And even the plague organism, as Steve said, we can track to Neolithic, skeletons that have been unearthed in the last 5 years, that these, the progression of plague, where the progenitor was mildly pathogenic, either by acquisition of genes, bilateral gene transfer, expressed out of context, or loss of information. You rapidly went to the most lethal bacterial pathogen, on the planet that we estimate has killed 300 million people. But this is all through, again, taking DNA, move it into a different context, or loss of information. That's really the surprise. And that's true for all pathogens. Cholera makes a very potent toxin that disrupts our, salt balance in our intestines. But that toxin is carried on a virus that infected the Vibrio bacterium at some point in its history. And that toxin may serve a very important purpose in regulating salt concentration in crabs in estuaries where the tides are coming in, the salt concentrations increasing and then decreasing. And for these crustaceans to survive, they have to be able to regulate loss of water or intake of water, turgor of the cell. And cholera toxin seems to play an important role in that process. So, but then you take it out of context and we eat that crab that has that bacterium that's regulating salt, then it's be very bad for us. So I think, botulinum toxins are on viruses that infected bacteria, diphtheria toxin. So we're really catching the infection of infected bacteria in these processes. So as Steve said, it wasn't part of the aboriginal design of these bacteria.
Let's go to some questions from the audience. And Jorge, you can come on now and select a question. Before I do, let me just get Steve's insight on what Scott just said there. Do you want to piggyback on what he just said there, Steve?
Well, we should, in fairness, come back to your original question about bacteria or about viruses now, because an interesting possibility, hypothesis, would be that viruses had an original, some kind of positive role originally in the repair of information, and we see hints of that in what's going on in the bacterial world. We know that we have viral insertions in our DNA, and so based on what we've learned about bacteria, and some of this has been learned fairly recently, that virulence is the result of information being expressed out of context or the loss of information. We might expect something similar now to arise as we discover more about viruses, that they may have had an original role in repairing information, and now we have information repair systems that are being expressed out of context, causing harm to organisms for which they were not specifically designed. So some of the things that Scott and I had just a fascinating conversation about this a couple weeks ago, and so there are things being learned about, for example, that viruses in bacterial populations can actually repair the original genetic endowment of the bacteria. So that's an intriguing thing to explore and something we're interested in. We know less about viruses than we do about bacteria, but the general idea, the general response to the problem of natural evil is to say, let's distinguish original design, aboriginal design, from subsequent decay. And viruses seem to be part of the decay equation, and they certainly are information that's being expressed out of context, and that's the indicator, I think, of something that's not as it was originally planned to be.
Well put. Thank you. All right, let's get some questions from our viewing audience here. Jorge, what do you have for us?
Well, first of all, I want to thank everybody in Facebook, Twitter, and YouTube that is watching this stream. Make sure you leave your questions. We got plenty here. Sam asks, what are 3 to 5 facts or arguments for intelligent design that are easy to comprehend and explain to somebody else?
Oh, very good question. Well, I think the first is the one we've been talking about, that at the foundation of life in the DNA molecule, there is a 4-character digital code that is responsible for producing the proteins and protein machines that cells need to stay alive. And we know from our experience, our uniform and repeated experience, which is the basis of all scientific reasoning, that information comes from an intelligence, whether we're talking about the information in a computer program or information in a paragraph in a book or assembly instructions or information embedded in a radio signal. Whenever we see information, we always come to a mind. It has always come from a mind, not a material process. So the discovery of information at the foundation of life is decisive evidence for a designing mind in the origin of life. That's one big argument. A second one is the argument from fine-tuning. If you look at this is something that physicists have been discovering since the 1950s, that the basic laws of physics and the initial conditions of the universe were set up just right to allow for the possibility of life within very fine tolerances. So you have these, for example, the strength of the gravitational attraction or the expansion rate of the universe. If any of these factors were off by a little bit, a little stronger, a little weaker, a little faster, a little slower, then life would not be possible in our universe. So physicists sometimes talk about our universe as a kind of Goldilocks universe where everything is just right. But as one great astrophysicist, Sir Fred Hoyle, put it, he said that a common sense interpretation of this evidence suggests that a super intellect has monkeyed with physics and chemistry to make life possible. So the fine tuning of our physical universe is a powerful evidence for intelligent design. And then the systems that Scott works on, not necessarily the virulent ones, but the aboriginal systems, these beautifully designed molecular machines, provide evidence of a kind of master engineer at the foundation of life. So those are three. I'm writing about others in my book on the return of the God hypothesis. I think another powerful evidence that is not so much an evidence just for intelligence, but for a transcendent form of intelligence, is that the universe had a definite beginning. Mm-hmm. This is something that's come into our awareness through astrophysics and astronomy over the last 100 years, as scientists formulated what was first called the Big Bang theory. And this is simply stunning that the material universe, if you go back far enough, reaches a singularity, a point where matter, space, time, and energy themselves come into existence. And as a consequence, there's really no material explanation that can be posited because it's matter itself that comes into existence at a finite time ago. Even time and space apparently begin. Mm-hmm. And so this is something that you would expect on a theistic view of the world. In fact, if you're a Christian or a Jew, you'll remember the first words of the Bible, in the beginning. And the modern astrophysics has discovered that there was in fact such a beginning. And so I think that's another thing to put all of this evidence in context. You have evidence of design, but also evidence of design from the very beginning of the universe and the fine tuning, and evidence of a beginning in these discoveries from astronomy and astrophysics.
Excellent. I got another one here by Levi James. He says, people continue to say that the coronavirus is basically equivalent to the common flu. Just how serious is this virus, and is it worth causing all this global pandemic?
Scott, what do you think?
I think, it's still up in the air. The mortality rate of influenza is about 0.1%. Okay, so 1 out of 1,000 people are going to die. Coronaviruses are maybe 2 to 3%. We're not sure yet as it plays out. But the coronaviruses that we've encountered in the last 2 decades, SARS in 2003, that mortality rate was around 7 to 9%. And it was shut down very quickly. MERS, the Middle Eastern respiratory virus, has an even higher mortality rate, and these are very closely related viruses. So I think, it probably wasn't wrong to err on the side of conservancy to see how this was going to play out. It's not maybe as deadly as was initially considered, but nonetheless, there was precedence for that deep concern. And look at this pandemic, how rapidly it's spread from China all over the world.
It seems to me, I've mentioned this earlier, that we know more about the morality of the coronavirus than we do the science. We know it's wrong to indiscriminately let a virus kill people when you can prevent it, but we're still unsure, as you just said, Scott, as to how dangerous is this, because we just don't have enough data yet. So we know more about the morality than the science at this point, despite the fact you always hear people talking about how great science is, and it is, but without morality, we wouldn't even be concerned about this virus, would we?
Right.
There was an interesting article in the Wall Street Journal about 2 weeks ago by 2 Stanford med school profs, Aaron Ben-David and Jay Bhattacharya. And they've made an interesting point about this issue of the mortality rate. And of course, the point they make is that the mortality rate is not the number of deaths divided by the number of confirmed cases, but it's the number of deaths divided by the number of total cases. As yet, we do not know that denominator, but there are ways of sampling the population that will give us a better sense of the infection rate, which would then enable us to calculate that denominator. And there was a study of a German village over the weekend where the infection rate turned out to be 15% of the population, which is an enormous number of people within a given population. And that infection rate computes to a 0.37 mortality rate for the local population based on the number of cases that are also in that area. And a similar rate of infection here would compute to about 0.09%, which is slightly a tick below the lethality rate for the flu. Now, we just don't know yet how widespread the infection rate is, but a curious paradox of this is that if more people are infected than we realize, then the death rate, the mortality rate would actually correspondingly decrease. But I agree with Scott. I think caution was the correct approach. But we need more sampling and more testing to figure out exactly how lethal it is. It may turn out that what we have is a highly infectious disease that is lethal at about the same level as the flu. It may turn out that it's higher than the flu. It may turn out that it's even lower. We really don't know. But so there's a lot of things we don't know yet. But a key thing to watch is this issue that these Stanford med profs highlighted. How infectious is it? How widespread is the infection? Because that's going to be an very important point in public policy to decide, what we do, because it will determine the death rate. And a number of the projections that have been made about, for example, the number of hospital beds that have been needed, and these have been thankfully overshot by quite a ways what's actually been needed, even in places like New York. So we do the best we can on modeling, and then we have to adjust the models as we get more data.
Got another one here. Afrin asks, is it possible to reverse engineer a virus? Could it be a real possibility that this was designed in a lab?
Scott, what is that possible?
Sure, it's possible, and I think there are the conspiracy theories, circulating that this could have been designed in some bioweapons laboratory, but I think it's highly improbable. But yeah.
Okay.
There was an interesting study in London by some scientists who examined the virus and concluded that it wasn't engineered. But that's interesting because it implies that you could detect design if it had been, and that's a premise of the intelligent design movement, that you can detect design. We detect it in the code, but there might be other ways to detect design. So it's just interesting that people have framed the question that way, design or no design, because that's something we've also asked about life itself.
Got a great question here. I think it's a bit personal for both of you, but I think our audience will be, built if you guys answer. Are you two locked out of the scientific community because of your beliefs in ID? How has that affected your careers?
I'd say that we've been able to create a new scientific community. It's been pretty exciting. If you look at what's happening internationally with the scientists who are involved in research on intelligent design, and we have groups in Israel and Germany and Brazil and many places around the globe. Our books are getting translated into all kinds of languages, and every year we have at Discovery Institute an extraordinary group of young scientists who come from all around the world to participate in our summer seminar. So yes, a lot of us, Scott included, have experienced opposition and harassment, hostility in their own departments, where our folks are outnumbered in their research environments. Locally, they're outnumbered, but internationally, we've developed a really exciting community of scientists who are working on these fundamental questions, and so I think for me, being at the Discovery Institute, where we're coordinating a lot of this, I get to see the big picture and see how many young people are being attracted to these ideas. I think we're attracting the young talent a lot faster than the other side. So we're very hopeful about the future. But Scott, Mike Behe, Bill Dembski, Doug Axe, Gunter Beckley, some of our leading scientists have taken a lot of flack, and there's been a lot of attempt to suppress the message, the argument. But I think it's getting out, and that's the truth will win out, right?
Right. Excellent.
Scott, well, Scott, has that affected you much? The animosity one might receive by being a proponent of ID?
Yeah, I mean, there's always, it's part of the territory. If you challenge a ruling paradigm, then there can be consequences, because people hold their ideas and concepts, very dear. If that's their worldview is built on this, then and you challenge it, then yeah, you can expect a type of opposition. And often it's indirect. So, but yeah, but we've all felt it,
Got one by Frank.
Scott's being very diplomatic in the way he addresses this, appropriately so, because he's. But what I think is exciting is the students that are being attracted to this. We now have people that have been through our summer seminar who are now tenured professors at major state universities. So that's the picture of how things change over time.
That's fantastic, man.
I think that, what I find interesting is that I'm very careful, when I'm teaching a course, in terms of promoting my own viewpoints. I don't teach intelligent design in a classroom. I know the difference between a pulpit and a lectern, but a lot of my colleagues don't. I mean, they'll come right out and say that, to students, your whole purpose on this planet is to replicate DNA. You're just a machine to replicate genetic information. There's no other point. And, that's a profound metaphysical statement to make. That's introducing your religion into the classroom. Yet I'm the one that was censored on my campus for not teaching this, but holding the views. And, and having to talk to the president of my university at the time. Yes, there's religion going on in the science classroom, but it's not coming from the Christians. It's coming from my colleagues. They just don't recognize that what they're doing. And so the playing field is not level in that sense.
Well said.
Yeah.
Scott's had a few what you might call Nicodemus in the night conversations, though, with colleagues who approach him and say, I respect your stance and I'm kind of intrigued with it. And could you tell me more? So he's pretty tough guy, ex-football
But they
player.
never say it publicly. Oh.
Yeah, never say it publicly.
Yeah. I say my colleagues sometimes have these moments late at night when I'm in my office saying, hey, how do you deal with this question? Or, and how do you deal with convergent evolution? So it's, but they recognize there's a cost that comes with some of these views.
He's right now in the witness protection program on my screen. We
had to do that because of what he said, Right now I got Rusty ask, if God created... Okay, so Rusty says, if God created the first cell, was it a perfect cell? Did God create the first virus? And did decay come from sin or evil?
I think the first cell was an elegantly designed cell, and I write about this in Signature in the Cell, just the not only the genetic information, but the sophistication of the information processing systems that we find in even the simplest one-celled organisms, to say nothing of complex multicellular organisms like ourselves. When I was writing Signature in the Cell, we had in our office an architect-level programmer from Microsoft who was helping my colleague Doug Axe write a program to simulate what's called the gene expression system, how the information in DNA it regulates the production of the proteins and protein machines that cells need to stay alive. And one day he walked into my office and he threw a book down on my table very dramatically. He's a big, tall guy with kind of a shock of wild scientist-looking hair. And he said, "Steve, I get an eerie feeling that someone has figured this out before us." And I said, "What do you mean?" And the book, I looked down, it had the title Design Patterns. And it's apparently a standard textbook for software engineers and developers. And he said, and he explained to me that a design pattern was a sophisticated means of storing or processing information. And he said, as I'm writing the code to simulate what's going on in the DNA and in the gene expression system, I'm discovering there are all kinds of recognizable design patterns that we know of through high-tech digital computing. You've got automated error correction inside the cell. You've got a spell check on your laptop. We've got something similar inside the cell. We've got automated error correction. We've got nested coding of information. We've got information layered on top of other information. We've got, and he just ticked off all these sophisticated design patterns that were necessary to understand in order to write code to simulate how the DNA is being regulated. And so we see except then he had one final line. He said, it's very similar to the design patterns we see in modern digital computer, except that it's being executed with an 8.0, 9.0, 10.0 elegance, anything beyond anything that we've ever done. So we still see powerful evidence of that aboriginal design at the foundation of life. And I think what's going on with virulence, whether it's in bacteria or in viruses, is evidence of the decay that we're told to expect in the natural world. Now, within the field of biblical theology, there are different concepts of what's called theodicy, how that deleterious effect on nature entered the world. Was it purely by human sin or human rebellion against God, or was there prior rebellion against God that also affected nature? And there are different theodicies, and we'll leave that for this discussion to the theologians. But the idea is that what we're told to expect biblically is those two things: decay as well as design as well as decay. And we see them both. And the Bible provides a backstory about how that happened. And biblical scholars interpret that backstory differently, but it has to do with rebellion against the Creator. And I think that's the Judeo-Christian understanding of why we live in a world that wasn't as it was first intended.
We only got a couple minutes left, so we've got to wrap it up here, but I want people to be able to go and find your work. So Scott, tell us where people can go to find what you're doing and the work that you're actually providing to the intelligent design movement, and your other work as well. Where can they go?
I think probably the easiest place, all publications, you can find on PubMed. So,
okay.
That's the NCBI website. You can just type in MNIC essay and publications are there. So.
All right. And Steve, you had mentioned some videos earlier. They're fabulous videos I've seen before. Kinesin and other videos. Where can they go to see that and your work?
Yeah, I just say, first of all, Scott is a frontline bench scientist. So he's got just marvelous peer-reviewed publications about these systems at the foundation of life, these molecular machines, molecular genetics, studies of virulence. He was sent to Iraq by the military looking for evidence of, bioweapons and chemical weapons. He's really a very impressive guy. He's not a writer of books. I'm a writer of books, and I'm a synthesizer of research of the type that Scott does and others in our movement. So my first book was Signature in the Cell, which you can find on Amazon. The second was Darwin's Doubt. And we created a website around the Darwin's Doubt publication. And on that website, we have playlists of videos, animations of these cellular systems that we've been talking about, the kinesin walking motor protein, the flagellar motor, the ATP synthase, the turbine that drives things. So all the things we've been talking about. A little video there called Journey Inside the Cell would be a great place to get a sense of the intricate design of the information processing system. And then a video, I think that's first up at the darwinsdoubt.com website, which is called Information Enigma. And that's something you can also find just on YouTube, Information Enigma. So those are ways into the work that we're doing on intelligent design. And we have a wonderful animator. He's in your part of the world, North Carolina. Big shout out to Joseph Condilas. He's an amazing guy, and he can bring these systems alive and open up by animating what's going on inside the cell. So a lot of the animations that we have were done by Joseph Condilas, and he's our secret weapon, because once you see the systems, you realize why the design hypothesis is so compelling.
Well, gentlemen, I hope you have a bunch of Nicodemuses coming to you as a result of this podcast, this live stream here. So as we move to the future, maybe people will say, well, I need to learn more from these gentlemen. So thank you so much for being here on the Hope One live stream. And we're going to do this again, I hope, soon. I also want to mention to our viewers, tomorrow, we're going to have Charlie Kirk on from Turning Point USA. If you want to see some positive events and movements going on college campuses, you're not going to want to miss Charlie Kirk. And on Friday, we're going to have Ray Comfort on to talk about hope through evangelism. So I want to thank Steve and Scott for being on today. Thanks, gentlemen.
Thank you.
Okay, thank you.
Jorge. All right.
Thank you, guys.